Good morning. The topics for today's session in kinesiology is muscle structure and function. So, what do you mean by a term muscle? Well, it is nothing but a tissue primarily composed of specialized cells which are capable of contracting in order to produce an effective movement, either with body or body parts with our effective external environment. So muscle has four main properties: excitability, contractibility, extensibility and elasticity. Excitability is ability of a muscle to respond to a stimulus. Contractibility is ability of a muscle to contract following a stimulus. Extensibility is ability of a muscle to get stretched without getting teared, and elasticity is ability of a muscle to return to its original position. Simple muscle contains many fascicles. Muscle fiber is composed of a cytoplasm called as sarcoplasm. The sarcoplasm contains myofibrils, which are the contractile structure of muscle fiber. This myofibril is composed of two filaments, thick filaments and thin filaments. This thick filament contains a protein called myosin and thin filament contains actin. So now we'll see about actin and myosin. So the structure of actin represents the helical structure where actin, troponin and tropomyosin, these three proteins present in this actin filament. So actin again consists of two parts, F-actin and G-actin. F-actin will bind with myosin and G-actin is a polymerase of small protein structures. Tropomyosin covers almost entire actin filament in resting state in the muscle. During contraction, troponin helps in changing position of tropomyosin, so the tropomyosin will release the covered structure so that actin will get bind to myosin and muscle contraction takes place. Troponin has three parts: troponin I, troponin T and troponin C. Troponin C gets attached to calcium ion, troponin T gets attached to tropomyosin and troponin I gets attached to actin. So now we'll see about myosin. Myosin also represents a double helical structure. It has two parts, a head part and the tail part. Head part has two active sites, one for F-actin to get bind to the myosin and another site is for the ATP to get hydrolyzed to ADP and phosphate ion during muscle contraction. Now we'll see about a contractile unit of a muscle. The basic contractile unit of a muscle is sarcomere. This sarcomere has areas that is either called as a zone or band which helps to identify this arrangement of thick and thin filaments. So the area where only the thin filament actin present is called as isotrophic band or I band, and the area where this thick filament is present, myosin, is called as A band or anisotrophic band. So another area such as M line represent only myosin presentation in the sarcomere, which is the center part of the sarcomere. And H zone is nothing but a central portion of thick filament where there is no overlap between thin and thick filament. So we need to consider tin, which is also a structural protein present in the muscle. It maintains the position of myosin. And another thing is a dismin, which helps in maintaining the parallel formation of the muscle structure. So now we'll see about cross bridge. For a muscle to contract, the muscle needs to get a stimulus, which is an electrical change taking place in the muscle called as action potential. This action potential causes the calcium ion to release, so the troponin gets attached to this calcium. Reposition of tropomyosin molecule takes place. Actin head gets attached to myosin head groups. This bonding of thick filament to the thin filament called as cross bridge. In this process, a tension is generated where hydrolysis of ATP causes ADP to form and phosphate ion. So in this downward image, you can see hydrolysis of ADP where myosin head is getting attached to actin. At the same time, ATP is converting to ADP and phosphate ion. In this left side image, we have given action potential propagation. So now we'll see about muscle contraction. So basically there are three types of muscle contraction. So muscle contraction is considered following the shortening and lengthening, how a thick filament is overlapping a thin filament and how the thick and thin filament are getting extended. In isometric contraction, there is a tension development takes place in the muscle length remains constant. In isotonic contraction, it is again divided into concentric and eccentric, where a muscle gets lengthened. The thick filament is pulled away from the thin filament. In isotonic contraction, we need to consider one thing: the tension develops in the muscle only occurs when the muscle resists the tension, so when the muscle resists the force from external stimulus, there is a tension buildup. The extension of this thick and thin filament causes this lengthening contraction. This concentric contraction and eccentric contraction. Another thing is the isokinetic contraction, where velocity came to play. So motor unit. What is a motor unit? It is nothing but a anatomical and physiological entity. Basic fundamental anatomical and physiological entity contains of single anterior horn cell and all the muscle fiber it innervates. So this motor neuron causes entire muscle to contract by firing asynchronously and repeatedly. So one more thing is this magnitude of the contraction may be altered by changing the number of motor units or the frequency at which these motor units get activated. Motor units vary according to their size, their axonal body, nerve cell body, axonal diameter, number of muscle fiber they innervate, and the type of muscle fiber they innervate. Each of this variation in the structure affects the function of a motor unit. One more thing is that small diameter axon takes more time for the impulses to travel. So we can see the small muscle groups where they preciously or accurately control the actions have small motor neurons, where the impulses take more time to travel through this motor neuron when compared to a large diameter muscle fiber which have a large motor neuron that takes time to travel. So this motor neuron again is divided into three types like our muscle fiber also. So slow motor neuron, fast oxidative motor neuron, and fast glycolytic oxidative motor neuron. So now we'll see about the muscle fiber. So we have three types of muscle fiber. Type 1 is slow twitch muscle fiber. Type 2A, which is an intermediate fast twitch muscle fiber, also called as fast oxidative glycolytic muscle fiber, and type 2A is a glycolytic muscle fiber. Where in type 1 aerobic metabolism takes place, in type 2 aerobic and anaerobic metabolism both will take place, and in type 2B anaerobic metabolism takes place. So one more thing we need to consider is a muscle having type 1 muscle fiber. More means they are called as stability or postural muscle fiber. These muscle groups compared to type 2, they get fatigue much lesser than compared to type 2 fibers. These type 2 fibers are designed for mobility purposes. They give larger range of motion for the bony component, so they get more fatigue faster and also take more time to recover compared to this type 1 muscle fibers. So now we'll see about architecture. So the two most important architectural characteristics that affect muscle function are muscle fiber length and the physiological cross-sectional area of the muscle. The muscle fiber directly determines the amount of shortening as well as the lengthening of the fiber. Consequently, a long fiber with more sarcomeres in series is capable of shortening over a greater distance than a shorter muscle fiber. The arrangement of muscle fiber and the length of momentum of the muscle affects the length shortening relationship. Both muscle fiber and the momentum must be considered. The physiological cross-sectional area is a measure of the cross-sectional area of a muscle perpendicular to orientation of the muscle fiber. The amount of force produced is directly proportional to number of sarcomere aligned straight by side. So we have unipinnate muscle fiber arrangement, bipinnate, multipinnate, fusiform, parallel and circular muscle fiber arrangement. So connective tissue. It is a covering of a muscle. So the fascia can be divided into two zones, superficial fascia and deep fascia. The zone of superficial fascia, which lies exactly under the dermis, is located directly under the derm, is composed of loose tissue contribute to mobility of the skin. The zone of the deep fascia is composed of compacted and arranged collagen fibers. This deep fascia attached to muscle and bone may form a tract or band and retinaculae. So we need to consider parallel elastic component and series elastic component. The parallel elastic component is the surrounding connective tissue of a muscle like sarcolemma, elastin protein, tendon and a nerve and blood vessel. These structures function in parallel to the muscle. Whatever the muscle do, whether the muscle shortens or lengthens, these structure works in parallel to this component. So they are called as parallel elastic component of the muscle. So the increased resistance of perimysium to elongation may prevent overstretching of this muscle fiber and contribute to tension at the tendon. So we need to see about what is epimysium, perimysium and endomysium. Okay, this is a covering of a muscle. A single muscle fiber is lined by endomysium. A group of muscle bundle is covered by perimysium and the outer covering is the epimysium. So in the next video, we'll see about muscle function in detail. Thank you.